Well, Nunkari Nungi, everybody. Welcome to this race grand round. My name's Paul Worley. I'm the Professor of Rural Health and the Executive Director for Clinical Innovation here in the Riverland Academy of Clinical Excellence. I'm on Ngarrindjeri country today and acknowledge the debt we owe to Aboriginal people across the country, but particularly on our country, for their stewardship of health, land and society. These grand rounds are an opportunity for us to engage with experts around the world, to learn and to contribute. And today we have the opportunity to learn from work that's being done in South Africa, in an area that we are looking at in our region. As many of you would know, in the Coorong region, we're just starting some research with Aboriginal people around diabetes remission. And it's a great opportunity, therefore, for us to learn from work that is going on in other countries. And we have an outstanding presenter today, Dr Amy Mendham, is a postdoctoral researcher with the University of Cape Town and Wits University in South Africa, who's been part of the diabetes work in South Africa. And it's a great privilege to be able to recognise that she will be joining us in Berry next month. In fact, she has already joined the staff of the Riverland Academy of Clinical Excellence as our Senior Research Officer. So with no further ado, it's a great pleasure, Amy, to be able to welcome you as a PhD and an exercise scientist to present some of the work that you've been involved with, and then to give us the opportunity to interact with you after that. Over to you, Amy.
Thank you, Paul, and thank you for the introduction. So today what I'll be doing is just presenting a small portion of the work that I've been doing since I moved to South Africa. I've been here for the last 8 years, and my main focus has been understanding the determinants of type 2 diabetes and the pathophysiology of the disease itself. Let me try and... Okay, now it's not progressing. There we go. So firstly, I'd like to make the acknowledgement of country and the traditional custodians whose ancestral lands and waters we are meeting on here today, and pay respect to the elders past, present and future. We acknowledge the deep feelings of attachment and relationship of Aboriginal people to the country. So firstly, I'd like to introduce you to the South African context. And coming from rural Australia, I was very confronted, I guess you could say, from what I was exposed to when I arrived in South Africa. So South Africa is it is the most unequal country in the world. So you have very extreme lifestyles of extreme wealth and extreme poverty living right next door to each other. And so since I've been here in the last 8 years, I've been working with people in vulnerable communities and people that have been living in the townships. So when you look in the townships, there's multiple contexts of living. So you have informal settlements and settlements that are all integrated with each other. So your informal settlement, I guess, is an example of the picture up the top, where you can see it's shacks, walking to get water, walking to toilets. And then you have your more formal settlement, which is down the bottom, which is more your permanent structures. So there's been 2 cohorts that I've been working with in South Africa, and they're the 2 largest townships there are. One's in Johannesburg, which is in Soweto, and it's known for the Southwest Townships. It was developed in the 60s as part of the apartheid regime. And then you have the one in Cape Town, which is in Khayelitsha, and it's translated to New Home in Azizi Khosa. And that is the largest township in the Western Cape. So within these settlements, you have about 75% that are living in informal structures. You have about 1/3 that have to walk to get water. 89% are moderately to severe insecure. You've got 46% of the youth that are unemployed. 73% have not completed their year 12, and then you've got around 70% of children who don't have access to early childhood development interventions like preschool daycare that prepare them for the schooling experience. So this creates a very interesting space. It's very complex and it's very intergenerational. So you've got a lot of obesity, poverty, unemployment that's leading into excessive disease risk, adoption of risky behaviors, emotional disorders, social cognitive impairment. And then obviously on what I'm most interested in, it's looking at obesity and metabolic diseases. So I think it's important that, like, although I speak about the pathophysiology of the Black South African community, it's a very complex space, and there's lots of socio-demographic characteristics and socio-demographic factors that interrelate to the findings that I'm going to show you today. So if we want to look at the prevalence of diabetes in the adult population in South Africa, it's the second leading cause of death, and it's the first leading cause of death in women. However, you're looking at about 85% of adults in this population that are actually undiagnosed. So you've got 13% of total have diabetes, but that's grossly underestimated given that the screening process and the amount of undiagnosis is so high. So what we're seeing in South Africa is that 20 years ago, it was the HIV epidemic. And now we're seeing this transition that the HIV is now being treated. People are living longer lives, and now life expectancy is increasing. And now they're getting this mix of communicable and non-communicable diseases that are interrelating with each other, which is also adding to the complexity of the disease development itself. So also to add to it, in the context of type 2 diabetes and risk factors, obesity is an obvious one that I'm sure we all know about. And the women in the South African population, you're looking at 68% are overweight and obese, which has increased significantly over the years, and then about 31% with men. So majority of the population that we're having most difficulty with regarding type 2 diabetes and obesity is within the female population. And when we look at understanding the pathophysiology and factors relating to fat distribution within the cohort, there's significant differences between the European population and the Black African population in where the adipose tissue is distributed. So the African population distributes, as you can see on the right-hand side, the majority of their peripheral adipose tissue around the hips and around the arms, whereas with the European population, it's distributed most peripherally. So when you think of it in your traditional sense that peripheral fat is protective and your central fat is at higher risk of developing disease, you would expect that the African community would be at a lower risk of developing diabetes based on looking at this fat distribution. However, they have a higher rate of insulin resistance and a higher rate of disease prevalence. So it started raising questions about what is happening in the adipose tissue, what's happening centrally in the visceral adipose tissue that could maybe explain the disease prevalence that we're seeing and the increased risk that we're seeing within these communities. So something that Julia Goodicke, who I've been working with since I moved here, she's been an amazing mentor for me. That this is a lot of her earlier work with Ann Sumner that she did. And it showed very clear differences in the European population, white South African population, compared to the African populations living in America, African populations living in Africa, and African populations who have actually migrated to America. And for the same waist circumference, your visceral adipose tissue is significantly lower in the African population compared to the white European population. And so for the same, so this immediately started raising questions on waist circumference cut points and how appropriate these cut points are in actually predicting the risk that we see within these communities. So a lot of the work that we've been doing and what Julia Goodicke has been doing over the years is to understand this pathophysiology and understand what factors actually relate to the disease progression that may not be so unique to the European population, but may be more unique to the Black African population. And I suppose this is what we came up with last year. And it was from data from longitudinal studies, cohort studies, cross-sectional studies, intervention studies. So we collated it all together to come up with a hypothesis on understanding the pathophysiology of the disease. So if you look at the context of the disease itself, the traditional, I suppose, start point of the disease is the increase in the peripheral insulin resistance. And that's been constantly, like quite consistently shown in the European population. But something that's quite unique that we are seeing within, especially within the Black South African women, is this hyperinsulinemia that's actually driving the peripheral insulin resistance. So it's the start point is actually different. And it's quite a consistent showing, and it's actually been shown in African Americans, it's been shown in adults and also in children. They're seeing in African American children, they're starting to hypersecrete insulin at a very young age, even before obesity develops. So our hypothesis in a very, I suppose, simple context is that your traditional factors such as your visceral adipose tissue and your central adipose accumulation may not be what's driving your peripheral insulin resistance and your overt type 2 diabetes, but rather there's something that's happening in the beta cells that's driving the hyperinsulinemia that is probably encouraging a lot of the obesity to start in the first place, and then creating a vicious cycle for beta cell failure and the development of type 2 diabetes. So that's essentially, like, if I could put everything that I've done in the last 8 years into one figure, this would be it. So it's something that's quite cool and that we put out there last year, and hopefully people can start trying to prove us wrong, I guess, in the context of good science. So our first problem, as I said before, was the waist circumference cut points. And I suppose you can also, like, if you look at other vulnerable communities around the world, you can see the little data that exists in these populations. So with respect to my presentation with the sub-Saharan Africans, the standard reference point was to use European data. And based on what we know and what I've just explained to you regarding the pathophysiology of the disease, it seems very inappropriate and to use these cut points in order to screen people within the community. And so it has driven us to start developing cohorts within Africa, which a lot of it was in the developed world, and now they're starting to try and build cohorts in Africa to try and understand the pathophysiology of the disease, and screening tools that can be used that are cheap, effective within the communities to help with the disease prevention. So Julia Goodicke, who's the lady in the middle, and Lisa Mickelsfield on the right, they started a cohort in Soweto, so Soweto's in Johannesburg, to understand the determinants in the Black African community, along with a lot of funding and a lot of investigators around the world. And I came onto this project a little bit later on, once the data had been collected, and they needed someone to help with the interpretation and help with data management and publication. So that's where I came into it. So the main purpose of building this cohort was to examine the sex hormone levels over the menopause transition in women and in men of the same age, and to explore the effects of body fat distribution and insulin sensitivity secretion when dissecting the roles of glucocorticoids and inflammatory markers, especially in the context of HIV. So this, as I said previously, this is, I suppose, has been driven by the increased life expectancy with HIV, understanding the interaction with HIV and menopause, and the interaction between the communicable and non-communicable disease. So this multi-morbidity type model. So to give you a bit of a background on the cohort, so it was started at baseline, the AUI-GEN cohort back in 2014, and that was around examining the genomic and environmental factors that influence body composition, fat distribution within the community. And then it was also combined with it's called the SWEET study, which is the study of women entering and in endocrine transition. So we had a nice number of men and women back in 2011 and 2014 that were then brought into follow-up part A and follow-up part B. So follow-up part A was 500 men and 500 women, because that's how much funding we got in order to support the data collection. It would have been nice to do more, but we'll get there soon. And these, for follow-up part A, they were heavily phenotyped. So they did oral glucose tolerance tests so that you could look at insulin sensitivity and insulin secretion, fasting bloods to look at all your classic risk factors for metabolic syndrome, and then inflammatory markers, hormones, androgens, and then also with what I'll present today is to look at the targeted proteomics profile as well. And then in addition to that, we're looking at body composition, physical activity, diet, mental health, stress. So it's a very, like I said, heavily phenotyped cohort, which we can use really nicely in follow-up data to look at incidence of type 2 diabetes. So what we also did as part of this was a subsample. So within the subsample, we looked at the menopause and HIV interaction, and we did more data collection on these women. And these women, like we looked at adipose tissue samples, so from the gluteal and abdominal tissue, to look at the pathophysiology of what's actually happening within the tissue, look at inflammation, fibrosis, and mitochondrial function and markers within these tissues to help like really go down into depth on understanding on the disease progression and the interaction that has with aging and HIV. So that gives you a nice background of the cohort. So I'm just going to present some of the outputs that we have now. There's a lot more to come because there's a huge amount of data, but I guess I'll be able to maybe present those to you in due course. So the first paper that was an obvious one to come out that Julia drove and Lisa drove as well was looking at different waist circumference cut points to predict dysglycemia and type 2 diabetes within the African men and women, and to compare these cut points to the European cut points based on the International Diabetes Federation. So if we want to look at these cut points, where I've circled now, these are the ones that the circumferences that we have identified within our cohort. And as you can see, the men are slightly different at 96.8, whereas the IDF cut point is 94 centimeters. Whereas the women, you can see there's quite a stark difference between the European cut point and what we are showing within the African cut point. And so with the cutoff of being 91.8. So the issue with this is also that it's also, although we have improved the International Diabetes Federation cut point, it's still not amazing. And it's probably still not one of the best models that we can use within the cohort. It's a good start, but I think there's a lot more room to move with building more cohorts and doing longer follow-ups to get a more accurate risk prediction model. So if we want to look at the main key points, we can show that the African waist circumference cut points actually differ to the European cut points. The African cut points perform better than the European cut points, although, as I said, they're still not at the sensitivity and specificity that you would like to have. And so larger longitudinal cohort studies are required to verify these cut points. within the population. And that'll lead into the next part of my presentation, as I'll explain soon. So this has kind of got us wondering what other things can we do and what other platforms can be used as risk prediction models that may improve the model for waist circumference, or may be used in addition to waist circumference, or maybe used in isolation. And we went searching, and I looked with our Swedish collaborators, and they brought my attention to this OLINK, which is like a targeted proteomics panel that can be used, and like they have inflammatory panels, cardiovascular disease panels, and some of them have your traditional markers, and then they also have your more novel markers that we can look within this cohort. And what they're actually doing with this OLINK is that they're bringing the pharmaceutical companies in now. So the UK Biobank is another cohort in the UK. Obviously, it's over 500,000 UK participants that they've been following up since, I think it's around 2008, and it's to understand the disease biology and development for effective therapies. So now the pharmaceuticals companies are starting to come in. There's 13 of them, and they want to build the world's largest scientific study on blood protein markers. And this is within, at the moment, 54,000 participants. And this got us interested on how that can be applied within our cohort. So I developed this paper from the MASC data that we had. We did the targeted proteomics and to identify convenient diagnostic risk prediction models that can be used in a clinical setting, and then also to help understand the pathophysiology of the disease itself. So to give you just a very general breakdown of how the data was managed, you have in the top left your discovery cohort, which is the MASC African cohort, and we split them up into normal glucose tolerant, impaired glucose metabolism, and then those with type 2 diabetes. We did the targeted proteomics panels, which was 184 targeted proteins. And then we also had oral glucose tolerance tests to look at insulin traits within the population. We looked at the relationship between the insulin traits and the targeted proteomics to find out what the relationship was between the protein itself and the disease endpoint. And we found 73 proteins were actually related to type 2 diabetes, which we then validated within the EpiHealth cohort. So these are probably, not sure how familiar you are with cohorts around the world, but the EpiHealth cohort is cohort in Uppsala. It's around 30,000 people that they've been following again over years. And we got in contact with Lars Lin, who is one of the PIs of the cohort, and he actually analyzed the exact same panels within just over 2,000 people. So we contacted him, and he was very willing to share the data with us. So we used his cohort as a validation cohort to find out how many proteins may have just been a false positive or a false negative. So we found out of the 73 proteins, you're looking at 34 of them that are European and African specific. But then there's also the 39 proteins that were African specific itself. And these also are the 39 proteins that I want to bring on and carry through further once we start doing more longitudinal follow-ups to see the difference between the European and African population and how these risk prediction models using these particular panels may differ between the population groups. So after we finished and looking at the data, and we were still looking at a lot of the hormone data, we wanted to know, because it's a constant trying to get funding, trying to work out where to go to next, so that we can follow up this cohort, because you don't want to let these cohorts disappear. And if you have no funding, then they will disappear. So in 2011, Julia myself and the rest of the team started looking for funding. We applied for a Wellcome Trust fund and we failed, which happens. And then we found it's called a PERMED, which is a personalized medicine grant, which we were successful with. And the main part of that grant was to integrate cohorts to identify personalized medicine that are unique to different areas of the world. So we put our thinking caps on and we started looking around at different cohorts that are already established worldwide. And we have a lot of collaborations in Sweden, so that was an obvious one, and then also in Ghana. So the Swedish cohort is the SCARPAS cohort. It's the cardiopulmonary bioimaging study. It's around 30,000 men and women. And then we've got the cohort in Ghana, which is part of the RODAM study. So it's a research on obesity and diabetes among African migrants. And it's looking at the Ghanaians that live in rural and urban areas, Ghanaians that have migrated to the Germany, Netherlands, and UK, and tracking these cohorts over a longitudinal context. And so we combined forces with the MASC cohort, the RODAN study, and the SCARPAS cohort to then create an opportunity to follow up on our MASC cohort in South Africa. So as part of the funding, it was an omics approach for personalized prevention of type 2 diabetes for African and European populations. So Julia Goodacre is the main coordinator, and our team on this side is Lisa Micklesfield, myself, and Dolly Ciponi. And then we have our partners in Germany and Sweden as well. So the main component of this project, and it's literally just been funded now, and the data collection will start, and it's to develop ethnic and sex-specific clinically feasible and cost-effective algorithms for early prediction of dysglycemia to inform culturally acceptable preventative nutrition therapy in European and sub-Saharan African populations. It's a bit of a mouthful, but it gives you a nice, clear idea of what our plan is. So just to give you a very basic breakdown, and then I've only got 1 or 2 slides left, of what is happening with this study, is that you can see our 3 populations. We've got our follow-up period, and over that follow-up period, so the Ghanaian population and the Swedish population have already been followed up, and then the South African MASC population is getting followed up as we speak, or hopefully that'll start soon. And with that follow-up, we're looking at incidence of dysglycemia. And when you're looking at incidence of dysglycemia, we're then looking at the targeted proteomics approach again to look at population and sex-specific early risk prediction, and then also to understand the pathological insights into dysglycemia in the African and European population. So the targeted proteomics study that I showed you previously, it was all cross-sectional. So one of the biggest limitation is cross-sectional studies are purely association, whereas if you can look at longitudinal studies, you can really look at the association in relation to the actual disease development and the incidence of the disease, which makes it a lot more robust models that we can use. So after we'll doing the targeted proteomics, we're also looking at untargeted. So targeted and untargeted then brings in your more hypothesis generating approach, where you look at the whole metabolomics profile, and you look at that in relation to dietary patterns to try and understand what type of dietary patterns within each cohort are actually relating to the metabolomics profile that are placing them at risk of developing dysglycemia later on in life. And so while managing the hypothesis generating approach and identifying clear nutritional patterns related to that metabolomics profile, you can then propose dietary modification for primary prevention of the type 2 diabetes within the cohort. So there's lots of steps within the study. It's the targeted approach, it's the hypothesis generating approach, and then it's also looking at the dietary patterns within each individual cohort and how that can be used as a modification and intervention profile within the communities as a disease prevention. And then the last part is looking at cost effectiveness and the willingness of the people within these communities to want to participate in a dietary modification program. And that will be the endpoint up until we get to like the personalized prevention strategies. So that this study at the moment, it gave us a really nice chance to follow up on the MASC cohort in South Africa. And it also gave us a really nice opportunity to integrate across multiple cohorts worldwide, which, like, to be honest, these are all things like Julia and Lisa and our collaborators worldwide. I've learned an extreme amount from these people in understanding how to integrate cohorts and the benefit of doing that. So I guess that's where we're up to now, and where I'm going to next. So we have the cohorts that I've been working with here, and then, as Paul mentioned, I'm on my way back to Australia in the next couple of weeks, ready to start my cohorts and my research there in understanding the disease and in rural communities in the Aboriginal population. So I'd like to thank everyone for listening, and then also the people that have helped and contributed to these studies, especially Lisa Mickelsfield and Julia Goodacre. Thank you.
Thank you so much, Amy, for that exposition. That fascinating work. And I noticed there was a gap in that world map, which was Australia. So how do you think that Australia can get engaged in this work? Because presumably there may be differences in parts of the Australian population in terms of the pathophysiology of type 2 diabetes and metabolic syndrome. How do you think Australia can get involved in this, and particularly the Riverland region that we work in?
Yeah, so can you see my screen still, or have I stopped presenting?
You've stopped sharing. Yeah.
Okay, cool, because I can't see you, but sorry. I think there's multiple vulnerable communities and Indigenous populations around the world, and I think it's quite nice because every context has different socio-demographic characteristics. There's different, as you could see, nutritional profiles within these communities, and so I think the world at this point is going to the direction of trying to integrate and understand the most that you can from what's already been done, and not actually having to reinvent the wheel, but just applying it in a different context. So I think in when you say with Australia and where we're looking at within the rural communities, a lot of what I have in mind is already been done. So it's just a matter of pulling it together and making it specific to the population that you're interested in. And so Australia can really benefit to, with these international collaborations and these international cohorts, from a methodological point of view, and then also from a physiological point of view, that we can then direct that's specific to Australia. I hope that answered your question.
Thanks. I'll open it up very briefly. We are running out of time, but if there are other questions, please raise your hand, or if you can't raise the hand, just ask your question. There is a question. It's from Tash's phone, but I sense it may be someone else. Tash, yeah.
Hey, how you going? Thanks for the presentation. That was great. Really enjoyed it. I missed the start of it, but I wasn't sure. Just pie in the sky stuff. So say that everything works out. What's kind of the dream? Is it to maybe have a screening test that school kids can do, or that people can get at the GP, or what's kind of what's the dream for you?
The dream is to have something a screening profile that is cost effective in vulnerable communities, because these communities, like in the clinics and like even rural communities worldwide, like rural communities in Canada, in America, in Australia, access to like your advanced screening is going to be very limited. So pie in the sky is to have clinical risk markers that are affordable within these communities that can identify these people early on so that we can intervene earlier.
Amazing. Thank you.
That's a fascinating question to look at how we contribute to that in Riverland Mallee Coorong, and I'm drawn to the work that Chris Palmer is doing in New York, where he's linking hyperinsulinemia and dysglycemia with mental illness, mental health, and with cognition in the brain energy hypothesis, which potentially with your work creates a linkage to how the social determinants of health are mediated in the incidence of diabetes, and vice versa. It creates a potential loop, which I think would be something that we could gain a lot from. Thank you so much for that. I'm sure there will be many other questions that people will have over time. And the good news is that we'll be able to have those conversations in person very, very soon. I thank everyone for their attendance today, and thank you particularly, Dr. Mendham, for getting up at 5:30 in the morning, Cape Town time, to be with us in our afternoon. Again, thank you to everyone, and to the Riverland Academy of Clinical Excellence team for coordinating these. Nakan, and we will see you at our next grand round.
Thank you, Amy. That was excellent.
Thank you. Thanks, guys.